So we are officially recording. And we had a question about the first one being calculating the length and the effective length, how RSM does that. So I do have a quick correction. Let me just pull up so you guys can see what I'm talking about here. So in this genes.results file, as well as, of course, we know the isoform.results file with the transcription, or with this transcript breakdown, we see in column 3 we have a length, and then in column 4 we have an effective length. So when I was thinking about calculating that length, the weightedness, for some reason I remember that it had to do with features from the reference. I was wrong. So this length here is actually weighted based on those isoform percentages. And so let me show you how that length is calculated. Let's go back to GeneLab book. Leave some stuff on here, guys. Sorry. So I'm just going to pull out the counts here just to show you the example of some stuff. Let me make this bigger for us. Look at. My computer's not telling. Okay. Let's make that a little bigger. Okay, so just looking here, I took the example of the gene that ends in 28 because that has 4 different transcripts or isoforms here. And so we see that the effective length was calculated, or the length was calculated to be 1863.69 base pairs. And so how that value was calculated is going to be based on the length of each one of these transcripts multiplied by the fraction of the transcript that's expressed based on the reads. And so if we take a look at that, so of course, if you guys can recall in the GTF files for each transcript, there's going to be a start and end position. Just bring up that GTF file again here. So in this GTF file, we have, in column 4, we have a starting location and an end location for each feature for each gene. And so that's how you can calculate the length of each one of these transcripts, as this is broken down by genes, and there's transcript names in here with the transcript IDs. That could be cumbersome, so you can also just look it up on Ensemble, which is what I did. Here it is. So I went ahead and I just looked up this gene, literally just Googled it, the Ensemble ID. And as you can see here, that's that gene that ends in 28, and then we see these 4 transcripts, the one that ends in 0028, 6990, 5585, and 1819 here. And so they give you a breakdown of the length and base pairs. So if you don't want to do those calculations from the GTF file to find out how long each transcript is, for most of these you can Google the Ensemble ID, and Ensemble will have a breakdown of how long each transcript is. So I kind of cheated there and grabbed that from there. And then with each one of those lengths, let me go back and pull that up. I'm going to take out my whiteboard again, guys. So with each one of those lengths here, so as you can see, can you guys all see this? Can anyone not see it? Awesome. Okay, so what I did here is I just have the gene on top and the length that was reported in the RSM file, and as well as the effective length, and then I'll get into that in a minute. And so we have the 4 transcripts that we have for this gene, and then this is just the length in base pairs that I put there based on that ensemble length that I just showed you guys, but of course that can also be calculated from the GTF file, and in fact that's how RSEM does it, because that's what we give them, is the GTF file. And then so I took the proportion of each one based on the isoform percentage. And just to remind you guys where that came from. So if you guys remember, after we took a look at the genes.results file, we took a look at the isoforms.results file. And then that breaks down and tells you just about the same information with an extra column here for each transcript for every gene. And so what we have here that I highlighted are the 4 transcripts for that gene that ends in 28. And what you can see is all the way on the right here, it gives you a percentage of the expression of each one of those isoforms based on the information you provided in the BAM file. So based on the number of reads that align to it. And so what you can see is that 70% of the reads align to that first transcript, 70% of the reads that align to a portion of this gene align to that first transcript, about 12% align to that second transcript, and about 17.5% align to that third transcript, and then we don't see any expression of the 4th transcript on this list for that gene. And so, of course, these are all in percentages, so to get the proportion, I just divided all these numbers by 100. And then that's how I got the isoform percent that's in parentheses here. And so if you go through and do those calculations, so multiply the length of each transcript by its percent expression in your samples for that gene, you'll go ahead and get values for the length of each one of these, or for the length of each one of these transcripts. And then you can sum it all up at the bottom. As you can see there at the bottom, and as you can see that number 1863, I got 0.66, I think that's just a rounding thing, matches the length for that gene. So that's how that length is calculated. Questions about how the length is calculated? Does that make sense? So essentially, I know that this says length, but it technically isn't an effective length because it's based on the weighted values of expression of each isoform. Anyone need that part explained differently or again? That's clear. Too early to do math. Okay. And then the next column that we have is the effective length. And so this one I know kind of tripped me up for a while, and I was trying to wrap my head around it. I had it before. But I guess it's been a minute. And so kind of going back and taking a look at different blogs and different forums, I wanted to find the clearest explanation that I could. And so, which one did I like here? I think it was a Google form that I liked. Yeah, right here. So I kind of like this explanation here. So I'm just gonna take it. So the effective length for the transcript is gonna be, it's a relationship between the length and the length of the transcript as well as the fragment, the size of the fragment. So when you go through and you chop up your original RNA, when you go and make your libraries, length question, how can one isoform be longer than the overall length? What do you mean? Okay, got you, Rashi. So you're saying, if we're looking at this here, and we're looking at the length of each one of these transcripts, you're asking how could one of these lengths be longer than the length that was calculated here? Is that your question?
Yeah.
Okay. So this length, I know it just says length here, that's a little misleading. This is actually a weighted length based on how much of each one of the transcripts are expressed. And so this transcript here, which also ends in 28, the same as the gene, that's going to be the transcript that contains all exons for this gene. And so the longest possible length for this gene's expression is going to be 2143. But then these other isoforms here, they are splice isoforms, right? So each one of these has fewer exons than the total number of exons possible, which is the number of exons expressed in this transcript. And so that's why you see that this transcript here that has some exons have been spliced out. I'm not sure how many, but ultimately once those exons are spliced out, we're left with this is the length of this transcript. And then in this transcript here, we have even more. So this is actually, the third one in the list is actually the shortest isoform of this gene. And so you can see that there are quite a few transcripts or exons being spliced out for this isoform. So it's really small. It's 832 base pairs. And then that last isoform, which actually isn't expressed in our samples, has a length somewhere between these two. And so when you go to calculate this length for the gene, it's based on how that gene is expressed in the data that we gave. And so this isn't necessarily only the longest transcript, because other transcripts of that gene could be expressed. And that's the case that we have here. So this is looking at expression of this gene for the sample. And so what we see here is that if you look at the isoform percentage, so it's a weighted average. So if you were to just take the average, like sum these up and divide by 4, you're not going to get this number. So yeah. Sorry for the dings. So it's a weighted average based on the data that you give. So for this sample, when RSEM went and mapped back the reads to a particular isoform using that maximum likelihood estimation, when it estimated that, what it found is that of the reads that are aligning to this gene in different regions on this gene, which of course covers all potential exons of this gene, what it saw is based on where those reads were aligning to, about 70% of the reads that mapped to this gene were identified to be coming from this isoform, and about 12% were identified to be coming from this isoform, and about 17.5% from this isoform, and then we got none of this isoform, this last isoform here, expressed in the sample. And so when you go to calculate the length of this gene, it's going to be weighted based on the percentage of each isoform that's expressed. And so that's why you take the length of each one of these isoforms, but then you multiply it by the proportion of that isoform that's expressed in your sample. And so you do that for all isoforms. And then when you sum up that value, that's going to give you this again, it should be weighted length here, based on the isoform percent in that sample. Any other questions on just this length column? Yes, no, yes, making sense, I hope. Okay, so now let's complicate things a little more and talk about effective length. And so it's kind of the form that I like. So effective length, what that one does is it takes into account the weighted length, but then it also says, well, wait a second, how much the total length of the transcript that you could observe is going to be dependent on the size of your fragments. So that's when we chopped up our original RNA, right? So we chopped up, there are of course copies of this particular gene and of each isoform of that gene. Obviously that's how we were able to detect it in our reads. And so how long our fragments are, so from the start of read run till the end of read 2 is going to be our fragment size, is going to determine how much of a copy of each one of those isoforms we can cover. And so when you do the effective length, your basically is put pretty nicely here by Colin Dewey. think he's one of the RSEM creators, actually. Is that true? Lauren, do you know that?
That sounds right, but I don't necessarily know.
That name sounds familiar. I think he might be the author of RSEM. So he's saying here is that the effective length for a transcript is essentially the number of possible start positions for a read or fragment within that transcript. Given that the read or fragment has to fit entirely within the transcript boundaries. So you have to make sure that the number of reads it could cover from start to, or the number of fragments from when you chopped up that could cover from start to end of a copy of each one of those transcripts, of course, doesn't go over the transcript length, right? It has to fit within that. And so that's why it's going to be based on how long those fragments are. And so, right, you can't have like a portion of a fragment. You have a full fragment aligning. And so kind of based on how that math works out, how they did it with RSEM, this is the calculation that they worked out to be able to determine how many, or how you can assess just how long each transcript you can actually cover based on the size of your fragment. And so usually that's going to be smaller than your total fragment length, unless you happen to have fragments that are all the same size, or the average of those fragments just happens to be perfectly divisible by the length of the transcript. And so the calculation that they use to determine the effective length is going to be the length of your transcript, depicted here by L, minus your fragment length, and then +1 for account for overhang. And so if we do that calculation, we need to take the average fragment length. Do you guys know where we get that information? We got to take the average fragment length for each read pair that's aligning to each isoform on this gene. Do you guys remember where that information is? You guys know which file? Any guesses before I bring you the answer? Of how would we find out the length of the fragment? So the length from the beginning of read 1 till the end of read 2 for all read pairs that align, big hint there, to that region, to that transcript. Any guesses where we find that fragment length? Yes, Inru, SAM file. And do you know what specific SAM file, since we're looking at isoforms and need to know the weighted isoforms, and of course isoforms are the different transcripts that can be made from a gene. Because remember, we produce two SAM files, which we output is the compressed version BAM files. We do one aligning to genome, and then the other aligning to transcriptome. So which one are we going to want to take a look at? Yes, Zinru, yes, transcripts. And so what we want to do is take a look at our alignment output for every transcript in that gene. And do you guys know which column we need to look at to find the total fragment length for each read pair that aligns to each transcript on this gene? Which column is that in the SAM file? Yeah, Rashi, column 9. So column 9 right here, the T length, that's the observed template length from the leftmost position of read 1 to the rightmost position of read 2. And so I basically did some Unix commands to go ahead, and what I did is I uncompressed the transcript file, which of course is in BAM format to get SAM format using SAM Tools View. And then I grepped So I used a grep command that I think we talked about once or twice when we were trying to troubleshoot something else. And I did that for each gene, and then I pulled out that column 9. But then because the first line for each transcript will represent one mate of the read pair, and then the next line will represent the mate of that mate pair group. And so what I did is I pulled out every other line, right? Because of course, this is a mate pair. So the total fragment for this read and this read is going to be 123. And so you just see opposite directions because, again, that's orientation relative to the reference that you're aligning to. So it's going to be in reverse based on if you have forward or reverse reads. And so when I did that, and I did it right on the, let's do here. So this is the command that I used. Those of you guys who are curious. So I did a SAMTools view of that sample that we were looking at, which is flight rep 1. And that SAMTools view, remember, will uncompress binary SAM files, which is what we have here, so that way we could look at them. And then I use this grep function to pull out each one of the transcripts. So here I have the transcript that ends in 28, and then I repeated this for the transcript that ends in 6990 and for the 58851 one and for the 1819 one. Of course, for the 1819 one, nothing came up because it's not expressed, which is what I expected. And so this is basically what it looked like here. So again, grepped that particular transcript, and then I piped it into this awk command here. Awk is a built-in program in Unix that I use all the time. And awk will allow me to print a specific column of a file. And as Rashi and Diksha pointed out, that transcript length is going to be pulled from column 9. And then I tried to pipe that then into another awk command, which will print every other line. But for whatever reason, it just got held up and took forever. So I went ahead and I just printed out that 9th column for that transcript, which again is the T length. So that's why you're seeing that positive followed by the negative number for each one of those mate pairs. So those are the transcript lengths. And then I just saved that in a text file, actually, and then just printed out every other line using that other awk command of that text file to get each one of the fragment lengths for every mate pair that align to that transcript. And then again, I did it for all 4 transcripts. And then to kind of keep track of everything, I actually put it all in an Excel file. So once I got this column, I just copy and pasted it into Excel. I know those of you computer scientists cringe at Excel, but remember, I'm not trained in this stuff, so I do know how to use Excel.
We don't cringe.
That's good. And so just to kind of blow this up here, what I did is I grabbed those numbers, so those fragment lengths for all the mate pairs that align to the transcript ending in 0028, and I just pasted them here. I did the same thing for the one ending in 6990, for 5585, and then of course, as we saw with the 0 expected counts for that last transcript, there were no mate pairs that aligned to that. And then I copy and pasted those fragment lengths and then just use the Excel function to take the average of each of them. And then I didn't know if this was also weighted based on isoform percentage at first. And so, because I couldn't find any forms where that was clear. And so I took the average of all of them too, and it turns out that didn't match the number. So that's how I'm assuming that these are also going to be weighted. And so now if we take those fragment length averages, so I want you guys to take a look at this bottom corner here. Sorry, my handwriting got sloppier as I went. So this is now that average fragment length from that Excel file that you're seeing. And again, I have it in the same order as I have up here, the transcripts. So I have the average fragment length for that first transcript. right here that you just saw in Excel, the average fragment length for that second transcript in the list from Excel, and the average transcript length from that third transcript in the list. And then what I did again is I took those proportion based on the isoform percentage that are expressed in our samples and multiplied it by this average length to get these numbers. And of course, that last transcript in the list isn't expressed. And then again, I summed up that to give me the fragment length size, the average fragment length size that's weighted based on the expression of each transcript. And I just put that number here too, so we could keep track of it. And then over in this bottom corner here, using that equation that I just showed you from the author of RSEM, is the effective length is going to be equal to the length of your gene, which again is weighted based on the length of each one of the transcripts. That's what we just calculated here. So that's what we just calculated down here, which, matched pretty closely to here with probably some rounding differences. And then what we see here is we take that length, that 1863.69, subtract our average fragment size, which again was weighted based on the expression of each one of these transcripts, and then add 1 to that to give us our effective length, which is 166.4.6, and again, pretty close. The effective length actually calculated by the program was 166.4.23 for this gene, for this sample. And so again, I assume that that's probably rounding. And so that's how that effective length is calculated. So I think, Felix, you were the one that asked this question. Does that explain how those numbers are calculated? I know it's kind of a lot of math for early in the morning. Any questions on either the length, which again is weighted based on the abundance of each transcript in your sample, or the effective length, which takes into account, could you post this photo on the drive? Yes, I can. I'll take a picture of this, post on the drive. I took a picture of my other whiteboards too, so I'll post those on the drive as well. So yeah, if you guys rewatch this or during one of the breaks and have more questions, or let that sink in after your coffee has taken effect, if you guys want to come back to this, if you have more questions, I'd be happy to answer them, because I spent quite a bit of time trying to wrap my head around this to explain it to you guys today. Any other, so if you guys have no more questions about that question, at least for now, I want to get into the other thing that I mentioned yesterday. And I was thinking about this a little bit wrong. So remember when we were taking a look at, bring up my notebook. Remember when we were taking a look at the total number of IDs, ensemble IDs, gene IDs, and it was over 55,000? And we kind of looked at the total number of lines in our GTF file. The population here. Somewhere. Scrolling up to the GTF file. Can we take a look at it? It's okay, we can look at it again. So when you look at the total number of lines in your GTF file, which I know we looked at somewhere, but I can't seem to find it. So looking at that, again, we could use that cat command to open it up. And then I talked to you guys about that function of WC-L to count all the lines of a file. And so as that's running, it's opening up that GTF file, the annotation file that we use, and it's going to tell us the number of lines. And the number of lines is actually quite a bit larger than, well, a lot larger than the number that we end up with when we create the counts table. Wait for that to finish. Okay, rather than sitting here waiting for that to finish, I did it on the actual command line. So this is on my local computer. And so I'll run that command here, make it a little faster. So if we take a look at the number of lines in that annotation file, we see that we have just under 2 million lines there. But then if you actually look at that like we did yesterday, so piping it into head, what we can see, I guess not so obvious here, but the gene IDs are repeated in this file, and those are indicated in that GTF file. They come after gene ID. And so I took advantage of the consistency of the GTF file and the location of the information of each gene in that annotation file. And so what I did there is then, let me make this cleaner. And so looking at this annotation file, we see gene ID followed by the ensemble ID for that specific gene that's represented in that line of the annotation file. And so what I did is I found some awk commands to be able to pull out specifically that term gene ID and print the phrase that comes right after it, which of course is going to be the ensemble gene ID. And so when you do that and just pipe it into the head function of awk, which I have here at the end, what you can see is that there are repeated lines or repeated ensemble IDs in those lines of the GTF file. So you'll have multiple information stored in different lines for the same ensemble gene ID. And so when you do that, what I wanted to see is, okay, well, if we have this repeated, there's almost 2 million of them. If you guys remember the counts table that we generated at the end of the FastQ to Counts Jupyter Notebook, we took a look at the shape of that or the dimensions of that, and we see a number here of 55,487. That's the total number of gene IDs that we see represented in that counts table. So in this first column here, all the rows are each gene ID. And then, well, I wanted to see, okay, that's obviously way less than the almost 2 million lines that we have. So it must only be taking all the information from one ensemble gene ID. And so in order to do that, you could use this other function called sort, another function of Unix, and then I can add a flag and sort called -u, and that says, go ahead and sort this, and then after you're done sorting it, only take out the unique lines. And so that's what I did here. And so if I just want to pipe this into head real quick, what you notice is when this first thing that I ran here, it doesn't do any sorting. And so that's how you see those repeat gene IDs. And so this command here, now I'm going to sort it, and I only want to pull out the unique ensemble gene IDs. And so you'll do that eventually. Probably should have cleared my screen. That's okay. It's because it's sorting through 2 million lines. So what you're going to see at the output of this head command is that we now have unique gene IDs. So I'll let that run. This is why I always get impatient and then just start running things from other places.
I think Phil's hand is up.
Go ahead.
Yeah, just a comment while we're waiting for the sort to finish. In case anybody's anxious about all these Unix commands that you might not have seen before, and this vertical line symbol for piping, just want to take a step back and explain to people what just happened. So Unix gives you all these command line tools to help you extract what's interesting to you from a data file and then present it in a way that's useful to you on the screen. And it's just text, but it's awfully useful. Sed and awk, I believe, are the two most complicated commands that ever show up in this kind of line, which is probably why we don't go into them. It would take another day. But I urge everybody at some point to pick up the superpowers of knowing how to do sed and awk. And the big deal here is this long, complicated line. Think of it as a pipeline. And everything in between pairs of vertical lines is another process on the data in the file, trying to get it first to extract and purify more and more pure just the information that you want and leave behind the information you don't want, and then to present it in a form that's going to be helpful. And in my experience, sort, of all the things you can put in a pipe, sort is the one that's most likely to slow it down. Because to sort n things, the amount of time is proportional to the square of the number of things. Everything else is pretty much linear. So that's why I figured I'd have time to say this while we're waiting for the sort.
Yeah, for sure. Thanks so much, Phil, for that additional explanation. Yeah, and guys, I didn't know how to do this before last night when I Googled how do I pull the phrase that comes after a phrase that I know. Literally, that's what I Googled. How do I Google a word after a known word, or how do I print a word after a known word in Unix? And that's how I found the command. But also, a lot of those functions that I do in Unix, there's Python ways to do it too. So a lot of you guys who are more comfortable in Python, you guys could probably figure out how to do this in Python. And again, I'm just doing this stuff to do some troubleshooting, right? Because I had made that statement that we have multiple gene IDs, ensemble gene IDs that are pointing to representing the same gene name or gene symbol. And so I had remembered running into that previously, which is why I mentioned it. And that number that I told you guys at around 25, 30,000 genes in actuality, I was thinking coding genes, so I'm sorry. There's more than just coding genes that are annotated, of course. So my fault for that. And what we have in this GTF file, again, so this here, what I did while Phil was helping further explain this command. So this, again, I'm just looking for the phrase gene ID, which is in the GTF file, and the word that comes after gene ID is going to be the ensemble gene ID. So that's what I wanted to do here. I wanted to pull out the ensemble gene ID in the annotation file, and I wanted to see that if I sort it, meaning that I remove any duplicate gene IDs, first I wanted to take a look to make sure that that's indeed what this function is doing. So I always check, each step of the command to make sure it's doing what Google told me it was going to do. And so just for checking this here, putting it into sort and piping it into head, we now see, whereas before we added the sort command, what we saw is we saw those ensemble ID repeats, right? And so that's probably why we were getting just over or just under 2 million lines in the annotation file, because there was multiple lines containing information for the same ensemble gene. And so I wanted to make sure that our final table here that we generated that we know has 55,487 lines, that's representing each unique ensemble gene ID. And so I went here and just added in another command that we talked a little bit about yesterday, the WC or word count command with the flag -L to count the number of lines. So now that I was confident here that I was pulling out the gene IDs, the ensemble gene IDs in the annotation file, in each line of the annotation file, and that when I sort them, I indeed now get all the unique gene IDs. Now I wanted to count how many unique gene IDs I had. So that's why I piped it into one more function called WC with that -L flag to count the number of lines. And what I see is indeed I get 55,487 total ensemble IDs, right? So that matches the number here in our table. But what I had told you is that I remember that some of these ensemble IDs match up to the same gene or gene name. And so I was way off in the number that I said. So the number I said is that there's only about 30,000 genes. I was thinking coding genes, but of course there are more than just coding genes annotated. We also have pseudogenes, microRNAs, long non-coding RNAs. There are several different types of other transcripts or RNAs that are annotated, and not just the coding one. So I'm sorry for that wrong number, but I still did remember that there were some that were mapping to the same gene symbol. And so again, just taking a look at the head. So now that we know that we have 55,487 unique ensemble gene IDs, and we're using the 101 release version. This is the syntax for the annotation file from Ensemble. So just taking a look at the head of that again, the next thing that I wanted to look at is the gene name. And so I look at a few more lines. There's the transcript name. And then I see I'm trying to find an example of where you could see the gene ID. Should be in each line. Oh, here it is, gene name, sorry. So the gene ID is, of course, the ensemble ID. And then in this annotation file, you'll also see for each line, it has the gene name. You guys are seeing where I'm highlighting here. So I'm highlighting this gene name XKR4. And so the next thing that I did is I used that same function, but instead of searching for the word that comes after gene ID, which will give me the ensemble gene ID, I looked up gene name and looked for the word or phrase that came after gene name. And you'll see that that's wrapped in quotes and a semicolon. So these set commands are just removing those things. And then again, I want to sort it because, of course, with the same exact ensemble IDs, we do indeed have the same exact gene names, which you would expect. But I'm arguing that we have, if there's multiple gene IDs mapping to the same gene name, should we have more, less, or the same unique gene names? Less, yes, Zinru. So if indeed what I had remembered and what I told you guys yesterday is true, if we are having some gene IDs that are mapping to more than one gene name, then we should see fewer unique gene names. And so again, I just ran this command here to make sure that indeed after this sort function, I am getting unique gene names. And so just while that's going, I'm also going to run that same command, but this time pipe it into that word count. And I'll let that run too while this is running. Any questions about my methodology here in terms of how I wanted to see if I was indeed remembering this right and remembering that multiple gene IDs can map to the same gene name? It's like you guys don't have to know the commands for this, but I do want you guys to be able to think about my thinking, my rationale behind doing what I did to try to sort out if I was remembering this right. Is anyone completely lost? Does this not make sense at all to anyone of what I'm doing and why I'm doing it? It's still too early for someone's thinking. So if you take a look at the output of this head command, I do now see that when I sort based on gene name, these are unique now. And so now what I did here is I grabbed that same command instead of piping the output of sorting the gene names into head to take a look at them. Instead, in this other terminal here, I told you I have multiple terminals up all the time, I am piping it into that word count command using that -L flag to count the number of lines. And again, this will tell us the number of unique gene names. And if indeed I was remembering this information correctly, then Zinru is right that we should see fewer unique gene names than we do unique gene IDs. And indeed we do. And so it's nowhere near the difference that I had mentioned yesterday. So again, I apologize for that. I was thinking of coding sequences, which of course are not the only genes that are annotated. And so as you can see, when we took a look at the number of unique ensemble IDs, that number is 55,487. When we look at the number of unique gene names in that same file, we see a less number. So we see a number that's less than the total number of ensemble gene IDs. So we see 55,367 unique gene names, which tells me that the only way that's possible is if more than one ensemble gene ID is mapping to the same gene name. Of course, it's less than I had thought, so it's only about a little over 100 ensemble gene IDs that have this issue. It's actually, when I was thinking of it, I was doing human stuff, and it's actually 1,000 roughly for the human in the same ensemble release. 1,000 genes that are ensemble IDs that are mapping to multiple or the same gene name. So it's not as dramatic here, but we still have over 100 ensemble gene IDs that are going to give us the same gene names. I know that was a lot. Any immediate questions about that? Okay, I will let Lauren answer the other question. I'm sorry that was so much, but you guys asked the question, so I wanted to show you how you go about answering questions like that you have. So most things you can answer with help from looking stuff up on the internet, as well as thinking through how you would figure out if your hypotheses on what's going on are indeed true. Lauren, I'll let you take it away, and then guys, you can kind of continue thinking about what we just went over, and if you have any questions throughout the day today, go ahead and ask.
Okay, great. Thank you. Thank you, Amanda. I have a much better understanding of what's going on now. Okay, so before we get into the rest of the statistics lecture, I wanted to go over a really high-level explanation of how we calculate principal components. So yesterday, Kevin and Yuval alluded to a whole bunch of linear algebra, which is true. The big idea behind principal components for PCA is simply being able to summarize all of the variance in the data set in several informative vectors. So what do I mean by that? How do we do that? The first step is to calculate what's called the covariance matrix between all the data points. So what that means is we talked about earlier how variance refers to how far a data point is away from the mean, how a data point varies from the mean. Well, covariance means if you take two data points, how similarly do they vary from the mean? Are they varying in the same direction? So they're both greater than the mean, or they're both less than the mean, or are they not covariant at all? They're totally different sides of the mean and far away from each other. So a covariance matrix tells us basically for all possible pairs of points, we compare all pairs of points in the data set. What is the relationship between each pair of points? Basically, are these points correlated? Then we use this covariance matrix to calculate what's called in linear algebra an eigenvector. And an eigenvector simply means the direction. If you plot your data on x and y axes, the direction of the axes where there's the most variance. And so I have a plot that perhaps some of you have seen before, and let me know if this animation isn't showing up on your end. But the idea is if you've plotted all your data points here on the y-axis, the x-axis, you can turn this line in all these different directions. And this line represents an eigenvector, this long line that keeps turning here. You want your first principal component to sort of stop wherever there is the most variance, wherever it will capture the most variance in the data set. So if you think about where's the most spread on the data set, well, the data points are going from the lower left up to the top right. That's the most spread in the data set. There's also a little bit of spread from the bottom right up to the top left, but it's less. The amount of spread is less in that direction. So in order to capture the most variance, the most obvious variance with the first principal component, this eigenvector line will the first principal component eigenvector will stop right here where it matches these two purple lines, and that will capture the most variance. And then the second principal component always needs to be perpendicular to the first principal component because of the assumption that generally if there's a lot of breadth, there's a lot of distribution in one direction, there's also going to be some distribution in the orthogonal direction, but it's going to be less. So the second principal component is this thinner line that you can see spinning here perpendicular to the longer first principal component line. And so we want to stop that line along the opposite axis. So that's how you get the first two principal components. And then you can imagine, well, there are also other sources of variance in other directions in this data set. So like there's a little bit of a spread in this direction because there are a few data points out here. So that could be the third principal component. And so you sort of iterate along these axes until the math is satisfied that you've captured the majority of the variance in the data set. So that's how to calculate principal components. Let me know if there are any questions on that. Yes, definitely, Amanda. I've seen that cartoon before, but it wasn't very well explained. And so it just served to confuse me more, but I found a really good explanation of it. Hopefully I conveyed it. All right, so if there are no questions on PCA, I'm going to move to where we were. I'm going to just skip back one slide because we're going to start talking about adjusted p-value. So I want to just review p-value really quickly. The p-value is simply the probability that a null hypothesis is true. If you're trying to establish whether or not you can accept your hypothesis, you need to first be certain that you can reject a null hypothesis. The way that we do that is to assign a probability to the truth value of your null hypothesis. And we do this using statistics, which I'll explain in a moment, but we generally accept a p-value of less than 0.05 because that means there's only a 5% chance that our null hypothesis is true. Now, in general, in bioinformatics, we use what's called an adjusted p-value rather than the straight p-value. So that just means we need to do a mathematical adjustment to the p-value before we can use it. And what I mean by that, and why we need to do the mathematical adjustment. Let's say Mel, our alien friend, performed RNA sequencing on the iris samples. It turns out, the mouse genome has maybe 55,000 genes. It turns out some iris genomes have over 100,000 genes. And in this case, what Mel is doing is each gene is a variable. And so RNA sequencing would give about 100,000 variables to compare between the iris species. So in this case, the question that Mel was asking, the hypothesis that Mel was testing was, is there one gene that can explain the difference between the iris species? That's the hypothesis. We're testing it. Well, we're testing it 100,000 times. So in one experiment, we're testing this hypothesis over and over and over and trying to establish the probability that the null hypothesis is true 100,000 different times. This introduces the problem of false positives. And we call this type of experiment multiple comparison testing. We're not just testing a hypothesis once, we're testing it thousands of times. So multiple comparison testing. This introduces false positives. So a way to think about why would multiple comparison testing introduce false positives in the first place. Let's say, instead of looking at 100,000 genes, let's just say Mel is looking at the difference in expression of just picked out a random set of 20 genes. Well, and then compared them between the iris species. By chance, we would expect that on average, 1 out of 20 genes would appear significant if we use the 0.05 or 5% p-value cutoff. And so you can imagine that in a data set of 100,000 genes, you're going to get a lot of false positives. So a lot of genes are going to appear significant just by chance. So because we're testing our hypothesis over and over on thousands and thousands of genes, we introduce false positives, results that appear significant, but in reality are actually due to random chance. In order to take into account the probability of false positives, we adjust our p-values using some mathematical and statistical adjustment. And the adjustment is dependent on the number of variables. So with high dimensionality data, the chance of getting a false positive goes up the more variables you have. So the adjustment that you do to the p-value has to change to take into account the increasing probability of false positives. So most bioinformatics statistical tools these days are designed for multidimensional data like RNA-seq data. And so they will generate for each variable first a p-value, which we know does not take false positives into account. And then they will also generate an adjusted p-value where they've mathematically adjusted the original p-value to take the chance of a false positive into account. So given what we've just talked about, what do you guys think we should use when we are determining the significance of a gene in an experiment where 100,000 genes are considered. Should we use p-value or should we use adjusted p-value, and why? Yeah, exactly. We want to use adjusted p-value because we have so many variables, we have so many genes. Exactly. Very well done. And the reason to do this would be because adjusted p-value takes into account the likelihood of false positives in a large multidimensional data set. Now let's ask the opposite question. When would it be appropriate to use a p-value rather than an adjusted p-value? You don't always have to use adjusted p-value. Let's say we have the p-value cutoff, significance cutoff of 0.05 or 1 out of 20. When could we use p-value instead? What do you guys think? So those of you who are saying smaller, smaller than what? Yeah, exactly. Veronica, yeah. Good thoughts, everyone. Technically, this you want fewer variables than 20, and generally speaking, it's what you all said. So you just want a smaller pool of variables. You have to think, well, there are few enough variables that false positives using a 1 out of 20 cutoff are not reasonably expected. So, yeah, good, well done. Now, the thing is, if we're using p-value to determine significance, and we go from p-value, and then we do an adjustment to take into account, well, some of these genes might be false positives. Which do you think is going to generate more significantly differentially expressed genes in Mel's RNA sequencing experiment? Is it going to be the p-value or is it going to be the adjusted p-value? Just how many, the number of differentially expressed genes that are going to come up. Yeah, exactly, Hanley. So yeah, p-value due to potential for false positives. Yeah. So exactly right. The p-value is a less stringent metric. The adjusted p-value is designed to hopefully remove any differentially expressed genes that were actually not differentially expressed and were just false positives. So the p-value is going to come up with more differentially expressed genes than the adjusted p-value. Great. Okay, so how do we actually calculate a p-value, though? So the way that we do this is given our data, we've done an experiment, we have our data. It doesn't have to be RNA-seq data, it just has to be data with a bunch of measured variables between two groups. We can use a statistical hypothesis testing method, and a simple hypothesis testing method is a t-test. So we're going to go over that one in detail, and then we're going to use a more complex hypothesis testing method in the RNA-seq experiment. So the t-test, just at its core, compares the mean value of all the measured variables between two groups. So if you're looking at 55,000 genes or 100,000 genes, you're going to compare the means 55,000 times for all the measured variables between whatever two groups you're interested in comparing. So I have a little graphic here to hopefully make this a little bit more clear. So in Mel's experiment, let's say Mel wanted to compare the expression of all the genes in Setosa versus the expression of all the genes in Virginica, and come up with how many genes are have significant differential expression between these two species. Well, the t-test will go gene by gene. So let's say right now we're looking at just gene X, one gene, and we'll take the distribution of all the expression values across all of the Setosa flower samples. That's here in green. That's the distribution in Setosa for gene X. And then look at the distribution of all the gene expression values for gene X in the Virginica samples. And calculate the means for both of those expressions. And here, remember, we use the Greek letter mu to designate the mean. So mu1 is mean expression of gene X in Setosa. Mu2 is the same in Virginica. And then the t-test calculates what's called a t-score, which generally can be thought of as signal over noise. More mathematically can be thought of as the difference in the group means over the variability of the groups. So there are two means, but we also have just the general variability of the groups. And remember here, standard deviation is coming in handy again. Sigma 1 is the standard deviation of gene X in Setosa, and sigma 2 is the same in Virginica. So a t-score is calculated using this equation for every single gene that we want to compare between the two groups, and we come up with the t-score. And then after the t-test, we get the t-score for each variable, and we know the greater the t-score or t-value, the more difference there is within that variable between the two groups. And also, the t-test calculates a p-value, which is associated with each t-value. And this p-value describes the likelihood of the null hypothesis being true for that variable. So in this case, the hypothesis that we're testing is gene X is significantly differentially expressed between these two iris species. The null hypothesis is this gene is not differentially expressed. So given the t-score, a p-value is calculated which describes how likely it is that the null hypothesis is true. So how likely is it that this gene is not differentially expressed between the two groups? So that's a simple hypothesis testing method. We can use it for RNA-seq data. We can use it for other types of data with multiple variables measured between two groups. Another and slightly more complex hypothesis testing method is called the walled test. Now, this is the hypothesis testing method that we're going to use with our RNA sequencing data. Now, the way that the walled test works is that it creates a mathematical model for the null hypothesis, and then it creates another mathematical model for the testing hypothesis, and then identifies which of these models best maximizes the likelihood of the data. So what do I mean by that? Well, first, a mathematical model is basically a description of a situation using math. So there are rules about how the data should behave within that situation. So we use general mathematical functions such as mean, standard deviation, and variance to describe if, for example, if this gene were differentially expressed, what would the data have to look like for that to be true? And you can just kind of visualize this as being, well, you know, the means of these two distributions would have to be far away from each other. That's one very simple mathematical model. It doesn't encompass everything, but it encompasses part of what the data would need to look like for a gene to be differentially expressed. And then a mathematical model for the null hypothesis could look like, the means aren't going to be that far away from each other. So for data to fit within the mathematical model for the null hypothesis being true, these distributions would have to overlap so much. So given these mathematical models with rules about how the data would look if the null hypothesis were true or if the testing hypothesis were true, then the Wald test takes a look at the data and says, okay, this mathematical model is the most, the data is the most likely result for this mathematical model. The mathematical model fits the data better. So let's say if the distributions are right on top of each other, the Wald test will take a look at the two mathematical models for null and testing hypotheses and say, well, these distributions are right on top of each other, so the data fits the model for the null hypothesis better. So for every single variable that is compared, the Wald test performs this analysis, generates the two mathematical models, and then tests the data against those models. And for each variable, generates a p-value with the probability that the null hypothesis is true in each case. So just to wrap up and make sure that we understand what we're getting when we run these tests, let's say Mel's final gene expression data has measurements from 100,000 genes from all the irises. If Mel performs a t-test to compare the two groups, how many t-values and p-values would be expected from this experiment? Yeah, exactly, Hanley. 100,000. Does anyone not understand why that is? All right, perfect. All right, so each gene is a variable, so we get a t-value and p-value for each variable. What if Mel performs a Wald test to compare these two groups? Wald tests don't give out t-scores, but they do give out p-values. How many p-values would be expected? Why do you think that it would be double, Hanley? What does everyone else think? And then we'll discuss it. Oh, I see two models. Okay. So let's just talk through this again. So yes, the Wald test is running two models per gene. But the goal of running those two models is to figure out which model is the best and therefore identify the probability that the null hypothesis is true. So you're still just asking the same question as in the t-test, just in a different mathematical way. So you're asking, what's the probability the null hypothesis is true? And the way that the Wald test does this is by running two math models. Yeah. So because you have 100,000 variables and the Wald test runs these models on every single variable, you get 100,000 p-values, one for each gene or each variable. Is that unclear to anyone? Is anybody lost with this? I'm happy to spend a little bit more time here if anyone would like me to. All right, great.
Wait, Lauren?
Yeah.
Hi, I was wondering if you could speak to, is one of these tests done more than the other then? Or if you had a data set, would you run t-test and a wald test? Or are there situations where it's more applicable because it's like gets us more valid results? Like how would you apply both of these?
Yeah, that's a great question. So the t-test is sort of ubiquitous, widely applicable, and sort of lowest common denominator a little bit. So it's not as specific as you can get with the wald test. So the great thing about the wald test is that you can customize it for data sets that have different distribution types. So because the wald test is creating mathematical model, you can inform it about, well, my data has a really high peak, or my data has a really long right-hand tail. So take this into account when you're performing the models. The t-test is not that flexible because the t-test is only looking at the mean. So for RNA sequencing data, which has a very specific distribution type, and we're going to get into that a little bit later, we want to use a more flexible model that can be more specific to the distribution, such as the wald test. It's not wrong to run a t-test on RNA sequencing data, and you can still get good and valid results. But depending on the type of question you're asking, you may want to just get more specific and more mathematically flexible results, and so you want to use something like the wald test. Does that make sense? Awesome. And we will get more into the distribution stuff in a little bit. Oh, you're welcome. All right. So now we've been talking about things sort of generally, what are some statistic concepts that build on top of each other, and just in the last couple of slides we've started getting into what do we need to do specifically for gene expression data? What are the statistics that are appropriate specifically in a gene expression data analysis? And one of the very important steps that we need to take with gene expression data is to normalize them for differences in read depth across different samples. So gene expression data often have different read depth levels per sample, where read depth refers to the total number of reads that come off the sequencer for each sample. And this is because when you do a sequencing run, you put your samples on an Illumina sequencer, for example, the number of total reads in a sequencing run is split up between the number of samples. So the read depth for each sample in a certain sequencing run depends on the total number of samples. So you can imagine that you could do a sequencing run on one machine with 4 samples in your experiment, and then you could take 6 samples and run them on the same sequencer, and those 6 samples will each have a lower sequencing depth than the 4 samples because the total reads for each run were distributed between more samples. Then if you want to combine those 4 samples with those 6 samples and do some analysis, you'll run into a situation where if you could have, for example, twice as many reads in one sample as in another sample. So we have a cartoon here illustrating this particular situation. So let's say this happened in Mel's experiment. Well, if Mel directly compares sample 1 and sample 2, without any normalization, it'll look like all genes are more highly expressed in sample 1. But we don't know if that's biologically the case, or if that's because sample 1 was run on a sequencer and got a higher read depth than sample 2. So Mel, and we also need to do this, must first normalize all the samples by their read depth before we do any direct comparison of gene expression values between samples. So before we can run a t-test or a wald test, we have to do a normalization step. So before we get into the way that we normalize data in DE-seq2, I believe it's time for a break. And everyone can come back at 10:50.